All right, we're going to dive into one of the biggest challenges in all of neuroscience. How in the world do we decode our thoughts and feelings just by looking at an fMRI scan? Well, scientists have a couple of key methods, and today we're going to break down exactly how they work and what makes them so different.
This question right here, it perfectly captures the problem of fMRI analysis. An fMRI scanner gives us this massive, four-dimensional movie of our brain, but it's incredibly noisy. You've got noise from breathing, your heartbeat, even tiny little movements. The actual signal we're looking for, the brain activity from a specific task, is super faint. It's that whisper we've got to pull out of the storm.
And this number? This shows you just how big that challenge is. That little whisper we're hunting for, it's a signal change of only 1 to 3%. That's it. When a part of your brain fires up, the change in blood oxygen that the scanner measures is just that tiny. The entire field is basically built on being able to find this microscopic little fluctuation time and time again.
So how have researchers traditionally done this? Well, the go-to tool for decades has been the general linear model, or GLM. You can think of this as the original hypothesis-first way to go looking for that signal. Okay, so here's the big idea behind GLM. It's what we call a mass univariate approach. Now, that's a fancy way of saying it runs a separate little test on every single tiny cube of the brain, a voxel, and it checks if that voxel's activity matches a pattern we give it ahead of time. The analogy here is perfect. Imagine searching a huge crowd for your friend, and you know they're wearing a very specific red hat. The GLM is like giving a picture of that red hat to thousands of security guards, and each guard is only watching one person in the crowd. And you just ask them, hey, does your person's hat match this photo? So, what exactly is that red hat in our fMRI world? It's this thing, the canonical hemodynamic response function, or HRF. It's basically a standardized, idealized model of how we think the signal should look, how it should rise and fall over about 16 seconds after some neurons fire. The GLM just assumes that all real brain activity is going to look like this one single predefined shape. And how does the GLM know when to look for that red hat? Well, that's the job of the design matrix. This is really just a schedule of your experiment. You tell the model exactly when a task started and how long it went on. The GLM then uses this timing to look for that ideal HRF pattern, but only at the moments it should be there according to your schedule. And this brings us to the crucial turning point. We've built this really elegant system based on one huge assumption, that the brain's response is always neat and tidy and looks just like our perfect little model. But what happens when reality is a little bit messier? And this gets to the absolute core limitation of the GLM. The research is clear. The brain's actual response is incredibly variable. It's different from person to person. It's even different between your own visual cortex and your motor cortex. And for some groups, like stroke patients, it can be totally different. So if your model is only looking for that one specific red hat, you're going to miss everyone wearing a blue hat, a green hat, or even just a slightly different shade of red. And that leads to false negatives. You end up missing real brain activity just because it didn't fit your perfect model. So if that model-driven approach has this major blind spot, what's the alternative? Well, that brings us to our second method, independent component analysis, or ICA. And this is a totally different philosophy. Instead of telling the data what to look for, you just ask the data to show you what's there. Right, so ICA takes a completely different data-driven path. It's a technique called blind source separation, which means it just sifts through all the messy fMRI data to pull apart signal from noise, and it does it without any predefined model. The analogy here really gets the idea across. Imagine a photographer at a party who has no guest list. Instead of looking for one specific person, they just take pictures of everything. Later on, by looking at all the photos, they can easily spot all the different independent groups of people who are chatting and interacting. That's what ICA does. It finds all the signals, brain activity, heartbeats, head motion, you name it, and it immediately separates them for you. So how does this actually work for a task-based study? Well, first, the algorithm just does its magic. It breaks down that whole 4D movie into dozens of separate maps, and each map has its own unique time course. These are the independent components. Now, this first step finds everything. Then, in a second step, the researcher takes that same design matrix, the schedule of the experiment, and uses it to sort through all these components. They just ask, okay, of all these cool patterns we found, which ones actually line up with my task? Okay, so now that we've met both contenders, let's put them side by side. let's really hammer home the difference here. This comparison really gets to the heart of it. GLM is model-driven. It asks a really specific yes or no question about a pattern you've already defined, and it asks it at every single point in the brain. Its biggest weakness? That rigid one-size-fits-all model. ICA, on the other hand, is data-driven. It asks a much more open-ended question. What are all the independent signals hiding in this entire data set? Its main hurdle is that it's just a bit more complex, and it gives you a bunch of components that you, the researcher, then have to interpret and sort through. And this quote right here from the source material, it just sums it all up perfectly. A single task might actually activate a few different but related networks in the brain. ICA has the power to pull these apart and show them to you as distinct components. GLM, on the other hand, might just average them all together into one big blob of activation simply because they all kind of sort of correlate with the model. It can really oversimplify what's actually going on under the hood. So whether you use GLM or ICA, you end up with a map showing these blobs of brain activity. But a map isn't very useful without labels, right? And that brings us to the final piece of our puzzle. Exactly. It's not enough to say, hey, look, there's a blob of activity right here. To make science work, to make it communicable and something other people can replicate, you need a shared map. You need a standardized system for naming the streets and neighborhoods of the brain, so to speak. And this is where brain atlases come in. They're basically reference maps that carve up the brain into distinct regions of interest, or ROIs. By overlaying their results on a standard atlas, researchers can say they found activity in, for example, Brodmann Area 44, and a lab on the other side of the planet will know exactly what part of the brain they're talking about. It creates a common language for everyone. What's really crucial to understand is that there isn't just one single map of the brain. Nope. Researchers use lots of different atlases, and each one is designed for a specific job. The Harvard-Oxford Atlas, for example, gives you broad anatomical coverage. The HCP Atlas gives you an incredibly detailed map of the cortex based on how it's all wired together. And there are even specialized arterial atlases that map out blood supply, which is super important for certain types of studies. So we've covered the what and the where. Let's wrap this all up by thinking about how you actually choose the right tool for the job. At the end of the day, neither method is inherently better than the other. The choice really comes down to your scientific question. If you have a very strong, specific hypothesis, like, I believe this exact spot in the brain will light up when people see faces, and I want to test that, then the focus power of GLM is a fantastic fit. But if your question is more exploratory, like, what are all the different brain networks involved during this really complex learning task, then the data-driven discovery power of ICA is probably the way you want to go. So I'll just leave you with this question to chew on. Are you a hypothesis tester, searching the crowd for a friend you know is there? Or are you a pattern finder, trying to discover all the interesting groups at the party? Because understanding that distinction, well, that's the key to unlocking the brain's code.